I have to stay like that casually.
పోతాయి మరి ఖరాబ్ అయి కూడా పోతాయి.
Screen. Document. Yes.
Page number, sir?
No, I am not following your textbook. That's why I don't know what numbers are we are following. You just check out the title, what title we are mentioned. I was mentioning, you may check it.
Which chapter?
I am not following your textbook.
Okay.
You have a textbook, na? Just
Yes.
You open the syllabus like this. So many topics are there. Then page number also there, and then check it what page number is we are present in the classroom. While you are running the classroom, then you may check out. Okay. This is your syllabus. Already we have done the cell biology, cell theory, difference between the prokaryotic and eukaryotic cells, and also we have completed ultrastructure of the animal cell in their classroom. And then structure and functions of the plasma membrane also over, and functions of the proteins also over from the plasma membrane. And then we are going to discuss about the structure and functions of the cell organelles like endoplasmic reticulum, Golgi complex, ribosomes, lysosomes, centrosomes, and mitochondria and nucleus. These subcellular organelles we have to discuss their structure as well as their functions. So plasma membrane is over in the earlier previous classroom, and then we are going to discuss about the first cell organelle. Cell organelle is, see here. So endoplasmic reticulum. You may check the endoplasmic reticulum in your textbook. Then you follow the your reader book. Okay. Today our first topic is endoplasmic reticulum. Okay. We are already discussed in the earlier class. Yes. Endoplasmic reticulum. So this endoplasmic reticulum is also single membranous cell organelle. Endoplasmic reticulum, endo means inside, and the reticulum is a fibrous, sac-like organelle, and this is a complex system. It is divided into different portions. This organelle is divided into different portions and vesicles and tubules. We are observing the different regions in the endoplasmic reticulum is distributed to the vesicles like tubules and cavities. So it is connected with the nuclear membrane. It is connected with the nuclear membrane, see something like this. And suppose this is the nucleus inside of the cell. This nucleus contains the two membranes. Actually, this is the inner nucleus membrane, and this is the outer nucleus membrane. In outer nucleus membrane directly connected with the vascular forms like this. These are the endoplasmic reticulum. This is the endoplasmic reticulum. So endoplasmic reticulum directly connected with the nucleus. So these endoplasmic reticulum also showing the sacs. These are the sac-like and vesicular like showing like this, and here also showing like this vesicles, and some strands. These are divided into so on like this. So this is the endoplasmic reticulum, and endoplasmic reticulum again divided into two categories. Depending on the presence or absence of the ribosomes. What is the function of the ribosomes? Ribosomes are embedded, attached, or associated with the membrane, plasma membrane of the endoplasmic reticulum like this. So here we are observing the ribosomes, Ribosomes are attached in some way, some area on the endoplasmic reticulum. So these endoplasmic reticulum we are called as rough endoplasmic reticulum. What is we called rough endoplasmic reticulum because of the endoplasmic reticulum is associated with the ribosomes. Maybe ribosomes are both types, two types. Generally, when you are going to the eukaryote cell, ATS ribosomes are presenting on the these rough endoplasmic reticulum. This is the ribosomes are again we are discussing the further classes. Then in this area, remaining area which is not associated with the ribosome, so that's why we are calling the smooth endoplasmic reticulum. So this endoplasmic reticulum is directly connected to the nuclear membrane and which is associated with the ribosome, which is called as rough endoplasmic reticulum or granular endoplasmic reticulum. This is granules, ribosomal granules are associated with the membrane of the endoplasmic reticulum and smooth endoplasmic reticulum. So these two portions are take the responsibility in different way. So these two parts are functionally different from one to another one. So we have to discuss about the what functions are there. So see here, yes, almost these endoplasmic reticulum is found in the almost majority of the cells, except some cells are exception is there. Eukaryotic cells except mammalian RBC. So in RBC cells, endoplasmic reticulum is absent and also egg cells. Gametes, egg cells it cannot found the endoplasmic reticulum. And based on the structure, already we have told you endoplasmic reticulum is there and what we are called smooth endoplasmic reticulum divided into two categories depending on the present and absent of the ribosomes. First one, rough endoplasmic reticulum. Rough endoplasmic reticulum. This type of endoplasmic reticulum presents the ribosome to attach the outer membrane of the outer surface of the endoplasmic reticulum, which is participating in the protein translation, protein synthesis. So this is about the rough endoplasmic reticulum, and which is contains the so many different types of the proteins, glycoproteins, and so many complex molecules also attached to the this rough endoplasmic reticulum, and which is actively participate in the protein synthesis. And pancreatic and liver cell and global cells and endocrine gland cells, which is the rich development. They are showing the richness because of these cells are frequently producing the more and more protein. That's why the rough endoplasmic reticulum is more present in the liver cells and global cells and endocrine cells and pancreatic cells. Okay, and smooth endoplasmic reticulum. Second one is the smooth endoplasmic reticulum. See here, smooth endoplasmic reticulum is not associated with the ribosomes and also not participating in the protein synthesis, and it is the place of the carbohydrate and lipid metabolism. So the smooth endoplasmic reticulum is take care about the carbohydrate and lipid metabolism. Okay, and these are found in the cortex cells. This smooth endoplasmic reticulum highly developed in the secretory cells because of secretory cells having the steroid hormones. The secretory cells secrete the steroid hormones and different secretions. So that's why these smooth endoplasmic reticulum highly developed in the secretory cells. And these cells are these endoplasmic reticulum, smooth endoplasmic reticulum take care in further fewer functions are there, adipose tissue and absorptive cells in the intestine and glycogen sorting cells in the liver and spermatocytes and WBC are found more in the smooth endoplasmic reticulum. And then modifications of the smooth endoplasmic reticulum, sarcoplasmic reticulum. In muscle cells, this smooth endoplasmic reticulum converted as a sarcoplasmic reticulum. Sarcoplasmic reticulum, which is participating in the formation of the tubulin. So these are the sacs. The sacs are filled with the calcium. Calcium is most important for the muscle contraction. So that's why in the muscle cells, these smooth endoplasmic reticulum converted as a sarcoplasmic reticulum. And the myeloid bodies are find out by the Porter, which is found in the retina, and also myeloid bodies are found in the nervous cells, neurons. So neurons, between the neurons and retina, we found the myeloid bodies. The myeloid bodies are converted as a smooth endoplasmic converted as a myeloid bodies in some cells, specialized cells. Then already we have discussed about these morphology of the endoplasmic reticulum. Generally, endoplasmic reticulum, they are showing the sac-like, cistern-like, and vesicular-like. So tubes, cisternae, and vesicles, which are they are showing the different 3 or more different types morphologically, and they are take care about the majorly 2 or 3 functions are there. The first function is the protein synthesis. First endoplasmic reticulum participating in the protein synthesis, and the smooth endoplasmic reticulum is take care about the lipid and carbohydrate metabolism. Got it? So this endoplasmic reticulum directly associated with the nuclear membrane, which is exist surrounding the nucleus, and it has taken the responsibility for the protein, some portion of endoplasmic reticulum taken as protein synthesis, and the remaining part which are taken as metabolism of the carbohydrates and metabolism of the lipids. Okay, and this is also participating in the packaging sorting system. Endoplasmic reticulum also sometimes participating in the sorting system, and the secretory cells, which is these smooth endoplasmic reticulum rich in the smooth secretory cells. Then we are going to discuss about the another cell organelle, ribosomes. What is the next topic is? Ribosomes. Ribose is soma. Soma means body. So ribosomes are made up with the nucleic acid, RNA. Ribosomes are made up with the rRNA and protein. The combination of nucleic acid, protein nucleic acid. The ribosomes are the factories of the protein synthesis, and these are made up with the what we are called made up with the RNA and protein. So ribosomes were first observed by the Bernard Claude and 1941, and also called as microsomes or protein factories. Ribosomes are the protein factories. Fallade named as ribosomes. Fallade was named as what we are called ribosomes, ribonucleoproteins, particles, ribonucleoproteins. So nucleic acid, ribonucleic acid and proteins, the combination of the ribonucleic acids and proteins. So see here, ribosomes are present in all eukaryotic and prokaryotic cells, except the mammalian RBC. So in RBC, RBC is the empty cell. RBC is the empty cell. Almost all the cell organelles are absent in the RBC. So these ribosomes are also absent in the RBC. In prokaryotic cells, the ribosomes are present in the cytoplasm, whereas in the eukaryotic cells, present in the cytoplasm as well as attached to the membrane of the endoplasmic reticulum, which means in prokaryotic cell, we have observed in the prokaryotic cell contains only nucleoid membrane, and there's exempt for the from the so many cell organelles. So that's why in prokaryotic cells, ribosomes are freely present in the cytosol, freely distributed in the cytosol. But in case of the eukaryotic cell, we have found out free ribosomes as well as the attached or bounded ribosomes. Already we have discussed in the endoplasmic reticulum. In the rough endoplasmic reticulum, these ribosomes are attached to the endoplasmic reticulum. So that's why we are called the granular endoplasmic reticulum. So ribosomes are freely present in the cytosol, and also bounded ribosomes are present in the endoplasmic reticulum in the eukaryotic cell. But prokaryotic cell, those are distributed freely in the cytosol. And see here, these ribosomes are weighed or denoted by SI units. SI units mean Stedberg units. These Stedberg units are using in the centrifugation. So these we are divided into different subunits. Every ribosomes are divided into different subunits, and subunits are contains ribosomal RNA plus proteins. So see here, when you are observing the 70S ribosomes, see here, this picture is clearly shows you how many RNAs are participating in the formation of these ribosomes. See here, clearly showing to you the picture. This picture is two types of the ribosomes are there. First one is the 70S ribosome. First one is the 70S ribosomes and 80S ribosomes. S means Stedberg unit. So 78 ribosomes and 80S ribosomes. Generally, the morphologically, two different types of the ribosomes are we are observed in the cell. 70S ribosomes are we are found out in the prokaryotic cells and mitochondria and chloroplast. Mitochondria, chloroplast are also having their own ribosomes. So 70S ribosomes are we are found in the prokaryotic cells, mitochondria and chloroplast. And 80S ribosomes are we are only observing in the eukaryotic cells. And these 70S ribosomes and 80S ribosomes divided into subparts. So one small subunit and one large subunit is there in that both of the ribosomes. So 70S ribosomes divided into 50S ribosome and 30S ribosome. 50 and 30S ribosome. 50, 30S and 50S ribosome. These are smaller subunits, and this is the larger subunit in the 70S ribosome. Here is the 40S and 60S ribosomes. 80S ribosomes are divided into two subunits. This is the smaller subunit that contains the 40S, and larger subunit which contains the 60S ribosomes. Again, in the 30S subunit, it contains the 60S rRNA. It contains the 16S rRNA and plus 21 proteins, which contains the two contents are there. 16S rRNA and 21 proteins are there in the 30th subunit of the 70S ribosomes. And in the 50S subunit contains 33S rRNA and 5S rRNA, and the 34 proteins are we are observed in the 50S subunit, 50S subunit of the 70S ribosomes. And here you observe it 80S In the smaller portion, small subunit contains 80s rRNA plus 30 proteins in the small subunit of the 80s rRNA ribosomes, and here the large subunit contains the 20s rRNA, 5.8s rRNA, 5s rRNA plus 40 proteins. 40 proteins we are observed in the 60s subunit of the 80s ribosomes. Here the common rRNA between the two subunits is 5s rRNA. See here, this 5s rRNA, here we are observing the 5s rRNA. So many competitive exams are, those are asking this question. What is the common subunit, common portion of the two subunits, 70s and 80s rRNA, which means the answer is 5s rRNA. 5s rRNA is common in the both subunits of the 70s and 80s ribosomes. And then these are the morphology. These are the morphology of the ribosome. And yes, so here is it. So these are the structure of the morphology of the subunits of the 80s and 70s ribosomes. So next we are going to structure of the ribosomes. Generally, these ribosomes are showing the smaller subunit and larger subunit, and which is contains the different regions. See here, every ribosome which contains the two portions, smaller subunit and larger subunit, small and larger subunit, smaller subunit and larger subunit. The smaller subunits are associated with the larger subunit, and this larger subunit contains the different regions. Larger subunit contains the different regions. See here, we are observing the three types of the sites we are observing the larger subunit, and these are P site and A site and E sites are there. P site and A site and S sites are there. So different sites are there depending on their function, and we are discussing in the further. And next, so functions of the ribosome. What is the function of the ribosome? We are already discussed in the earlier. So the ribosomal function is the major and most important function is protein synthesis. What is the function of the ribosome? Is protein synthesis. So ribosomes done we are called as factors of the protein synthesis, factors of the protein. So that's why ribosomes are participating in the translation. What is the meaning of the translation? Translation means DNA converted to the mRNA. There is a transcription, and transcription mRNA converted to the protein by the ribosome. That is called the protein synthesis. So these ribosomes are connected the polypeptide chain, linked to the polypeptide chain. Polypeptide chain means those are connected the one amino acid to another amino acid by forming the covalent band. A covalent band, which is known as peptide bonds. The peptide bonds are forming between among the different amino acids, depending on their codon, mRNA codon, and decode the mRNA, then ultimately produce the this protein synthesis. And so see here, ribosomes are contains the two parts of the attachment of the RNA containing direction. So finally, ultimately, the ribosomal function is the synthesis of the protein, which means mRNA converted as mRNA decoded as or converted as proteins. So that is the first function, and the second function is protection. Ribosomes also participate in the protective function, and by the mRNA transmits the proteins between the subunits of the ribosomes and is protected from nucleases. So similarly, new synthesized polypeptide chains are protected from the proteases. So proteases, what we are called proteases. Proteases means these are the digestive enzymes. Proteases means digestive enzymes. So digestive enzymes, protein proteases, digest the protein. The protease enzymes protected by the ribosomes only. So next next function is uh, what we are called okay. So these are the numbering folded. Okay, these are the functions of the ribosomes. Again here we found out the so many different functions are there in the endoplasmic reticulum. See here, mechanical support, renewability, transporting of the circulatory systems, this of the substances, intercellular impulse conduction, and formation of the nuclear membrane, glycogenesis, detoxification. Now, smooth endoplasmic reticulum also participates in the detoxification and protein synthesis, as well as protein synthesis means the rough endoplasmic reticulum associated with the ribosomes, then those are actually participating in the protein synthesis only. So these are the two cell organelles combinedly present in the cell cytosol and which is associated with the nucleus. Then will done the protein synthesis and uh, uh, participating in the different functions. Next, we are going to discuss about lysosomes. Lyso means lysis means digestion, degradation, or breakdown. Lysis means breakdown. Soma means body. So breakdown body is the digestive body or breakdown body, lysosome. It is a single unit membrane enclosed with a dense matrix. So lysosome, generally they are showing like this. This is the lysosome actually. Lysosome is covering with a single membrane and which contains the dense matrix. Here we have found the dense matrix. This dense matrix contains different types of the enzymes, fully filled with enzymes. That enzymes are participating in the digestion, digestive enzymes, hydrolytic enzymes. So that the hydrolytic enzymes are present in the inside of the lysosomes. So many different types of the lysosomes are found here. You see here different types of the lysosomes are found in the different stages of the cell body. Those are actively participating in the phagocytosis, phagocytosis, exocytosis, endocytosis. So already we are told in the earlier class. So lysosomes are take care about the different functions. See here, Christian Duduwe. The first of these lysosomes, Christian Duduwe, and named as lysosomes. The size is very low, 0.4 to 0.m microns. And these very different types of the lysosomes shape and sizes, their spherical, oval, and irregular vesicles. And these are contain the extracellular or intracellular digestion. So it carries intracellular digestion and extracellular digestion, and also it is participating in the autophagy. Autophagy means self-digest, self-eating. So damaged organelles also eaten by these lysosomes. And autolysis means self-suicide. So that's why lysosomes we are called as suicide bags in the cell. So suicidal bags. Lysosomes also act as a suicidal bags, and it is also called the uh, polymorphism. Polymorphism exhibit the polymorphism. Polymorphism means lysosomes are active parts in the metamorphosis or polymorphism. Polymorphism means poly mean many, morphosis mean form. Many forms. Lysosomes are showing in many forms like primary lysosomes, which are act as a storage lysosomes, which contains the storage of the hydrolytic enzymes, and digestive vacuoles, phagolysosomes. Digestive lysosomes which act as a phagolysosomes. This contains the phagocyted extracellular material formed in the fusion of the primary lysosomes, and autophagic vacuoles, autolysosomes. Second, we are also called the secondary lysosomes. These contain the lysosomal enzymes like seen in the liver disease starvation, and residual lysosomes. Residual lysosomes, the exhausted lysosomes in the residual body formed after the process of the digestion is completed by phagolysis. Contains the residual body. It acts as a residual body. See here different endolysosomes, phagolysosomes, autolysosomes, and exolysosomes. Those are divided into morphologically in the place of the cell. So they have done the different functions: primary lysosomes, storage lysosomes, digestive vacuoles like phagolysosomes, autophagic vacuoles or autolysosomes, and residual bodies. So lysosomes are showing in 4 morphological forms in the inside of the cell. And then we have to discuss about the functions of the lysosome. See here, 4 most important functions are done by the lysosome. First one is the heterophagy. The first one is the heterophagy. Heterophagy means, hetero means different, phagy means eating. Hetero means different, phagy means means which means eating. So the lysosomes are digested extracellular material as well as the own cell material, intracellular material when you damage the cell organelles. So which is called the outer. They are consuming the foreign body or outside food material, which is known as endocytosis. Endocytosis. What is the meaning of the endocytosis? Means engulfing the outside material through the plasma membrane, which is known as the endocytosis. And phagocytosis. Phagocytosis means again eating the outside material. And pheноcytosis. This is divided into pheноcytosis. Pheноcytosis means drinking the liquid fluid. So cell eating and cell drinking. Phagocytosis means cell eating. Pheноcytosis means cell drinking. So, and food material are included fluid material. They are engulfed by the plasma membrane and form the vascular form, which is known as the these vesicles are fused with the enzymes body. This is known as endolysosome, and this endolysosome then they are going to the heterophagosome. So heterophagy. The first function of the lysosome is heterophagy. Autophagy means lysosomal digestion of intracellular material. Lysosomes also digest or clear the debris of the internal material of the cell. So their own organelles, they are eaten by the cell own organelles, worn out the cell, and the dead cells Dead cells also digested by the lysosomes, and cell debris and storage food material. All these things are digested by the autophagy method. And next, extracellular digestion. These are digested the outside material through the secretion of the proteolytic enzymes or lipolytic enzymes, like in the spermatogenesis. After spermatogenesis, the cells are connected by digestion of the vitelline membrane. That is the process is filled by the acrosomal process. So they are called the acrosomal process. This is the sperm, and the sperm contains a knob-like end. The tip of the knob contains the hydrolytic enzymes. These hydrolytic enzymes are secreted by the lysosomes only. So these hyaluronidase enzymes, and so many different types of proteolytic enzymes are present in the tip of the sperm, and then which is secreted at the egg. Egg is covering with the vitelline membranes. The vitelline membranes are digested by this hydrolytic enzymes. So this is happening by the extracellular digestion, and also exocytosis. We are called the exocytosis. So many times, suppose exocytosis is found in the different cases in the metamorphosis and ripening of the fruits. So next, autolysis. Fourth function of the lysosomes is the autolysis. Autolysis by the own cells. They digest their own cells by the lysosome. It's called autolysis, which is found in the duration of the amphibian metamorphosis. The young ones are counted as adult. Organisms are counted as adult one by the removal of their unwanted organs. Suppose tadpole larvae remove their own tail, and then it's converted as a frag. Tadpole larva, the Syrian tadpole larva, and tadpole larvas are they are removing their own cells and their own organs by the digestion of the autolysis by the lysosomes only. So these are the so many cases we are observing the lysosomal function. So how many functions are there? Lysosomes have done the heterophagy function, autophagy function, and extracellular digestion, and also autolysis. So these are the functions are we are observed in the lysosomes. And then we are discussing about the another cell organelle. The cell organelle is centrosome. What you call centrosome? Centro means middle of the cell. Centro means middle, center region, and soma means body. So these centrosomes are found in the central region of the cell, and which is divided into two categories because of it play a key role in the cell division. So centrosomes play a key role in the cell division, which is discovered by the Van Beneden. Centrosomes are discovered by the Van Beneden and called as cell center. It plays an important role in the cell division, and eukaryotic cells, some lower plants like algae, fungi, biophytes, stems, and gymnosperms, it is present in the cell division, and present in higher plants like angiosperms, prokaryotes, and diatoms, yeast, and non-flagellated and non-ciliary protozoans. So non-ciliary protozoans, and some prokaryotic cells, the centrosomes are absent. And amoeba also absent the centrosomes because of we don't need to divide in a proper way. So that's why these are absent in the centrosomes. Structure. So see here, cytosomes are present in the near to the nucleus in the cytoplasm. Metazoan lies outside the nucleus, but in the protozoa lies inside of the nucleus. So centrosomes are cylindrical in shape and contain the two components. They are centrosphere and centriole. See here, this is the actually the structure of the centrioles or centromere. So centromere two blades, microtubular doublets are there, and it divided into spherical region, centrosphere, and also centriole. And see here, it is forming with the microtubules. 13 globular microtubules are participating in the formation of the centriole. The centriole, which is following the 9+2 pattern. 9 pattern and 9+2 pattern. So what is the meaning of the 9 pattern mean? 9 triplets. 9 triplets are distributed spherically in the center of the axis, which is known as centromere. So it is how many are there? 9 triplets. 9 triplets. Triplet means 3. 9 triplets means 3 globular proteins. So 3 globular proteins units are which is exists the 13. Every globular protein is contained 13 units, 13 units, and subunits A subunit, B subunit, C subunit there. These A, B, C subunits are which we are called the triplet. So these are 9 is there. 9 is spherically arranged in the central axis. So between 9 pattern we are observing the centromere, and the 9+2 pattern we are observing in the mesosomes like exoneme. Exoneme means cilia flagella. In cilia flagella we are observing the 9+2 pattern. So 9 doublets are observing in the surrounding, and 2 singulets are we are observing the central region. So this is the basal body. The centrioles are the we are also called the basal body. Also called the basal body, and which is participating in the tubular formation. So functions. The centriole is in the formation of the mitotic spindles during the cell division. Which spindles? Mitotic spindles are formation is done by the centrioles only, and it gives us to the basal bodies and chromosomes of cilia and flagella. So these are the bases are in the mTOC center. mTOC center means microtubule organizing center. mTOC center also generated from the these centrioles only. So which is participating in the formation of the flagella and cilia. And in spermatids, 2 centrioles are present in distal centrioles from the axial filaments of the flagellum of the spermatozoon. So these are the functions. 2 functions. Generally 2 functions are there. First function is the function of the centrosomes is formation of the mitotic spindle. Formation of the mitotic spindle in the cell division. And next, these are participating in the production of the basal body. Also, we are called the kinetosomes. These basal bodies and kinetosomes are the base of the cilia and flagella. So these 2 functions are done by the centrosomes, and the centrosomes are made up with the tubulin protein and the lipids and the ATP enzyme, and these are the present in the center of the nucleus, near to the nucleus and center of the cell. So that's why we are calling the cell center. Centrosomes we are also called as cell center. So this is about the centrosomes. So we have more time. 10 minutes is there. Then tomorrow we are going to discuss about the mitochondria. Today we are focusing on the three different cell organelles. Those are, first one is the endoplasmic reticulum. So now we recap the today's session. So endoplasmic reticulum is divided into two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum, which are participating in the protein synthesis because of rough endoplasmic reticulum is associated with the ribosomes. And then second one is the smooth endoplasmic reticulum. Actually, the smooth endoplasmic reticulum are participating in the, see here, these functions are done by the smooth endoplasmic reticulum. They are giving the mechanical support, permeability. Permeability means the protein which is fostering the osmosis and transporting of the circulatory system, and the synthesis of substances like various enzymes like triglycerides, phospholipids, glycolipids, fatty acids, cholesterol is synthesized in the inside of the these endoplasmic reticulum because of endoplasmic reticulum is the place of the metabolism of the carbohydrates and metabolism of the lipids. And intracellular impulse conduction also generated by these sarcoplasmic reticulum, and which contains the calcium ions. The calcium storage ions are found in the sarcoplasmic reticulum, which is participating in the muscle contraction and nerve cell, and formation of the nuclear membrane. Yes, already we have discussed the smooth endoplasmic reticulum is the place of the metabolism of the lipids and the metabolism of the carbohydrates. So the part of that, these smooth endoplasmic reticulum also participating in the formation of the nuclear membranes or cell membranes. Nuclear membranes or cell membranes. Glycogenesis. What is the meaning of the glycogenesis? Formation of the glycogen. Formation of the glycogen. The liver cells. What happened? Excess of the glucose is converted to the glycogen. So glucose converted to the glycogen, which is known as the glycogenesis. The glycogenesis function is done in the liver, so which is done by the place of the endoplasmic reticulum. And the very important function of these smooth endoplasmic reticulum is detoxification. So removal of the utilization of the toxic substances into harmless substances. So which is called the detoxification. The enzymes of the smooth endoplasmic reticulum plays an important role in the process of the liver, and substances like drugs, waste fatty acids, steroids, and poisons, and toxic material, drugs, and the chemical substances, harmful chemical substances, and all are detox. The alcohol, those are all are the detoxification is done in the liver cells. In the liver cells, the cells contains a smooth endoplasmic reticulum. So smooth endoplasmic reticulum is participating in this detoxification. So too many functions are done by the endoplasmic reticulum. In the part of that, first one is the protein synthesis. Protein synthesis is responsible by the rough endoplasmic reticulum, and smooth endoplasmic reticulum done the so many different functions already described here. And the second one is the ribosomes. We are focusing on the ribosomes, and the ribosomes are bonded. Sometimes we are found in the bonded, and sometimes we are feeling ribosomes we are found in the cells. And these ribosomes are major structurally. Two types are there: 70S and 80S ribosomes. Some 70S ribosomes we are found in the prokaryotic cells and mitochondria and chloroplast. And eukaryotic cells we are observing the 80S ribosomes. It contains the different subunits, large subunit and small subunit, and these are divided into 70S ribosomes are divided into 30S and 50S ribosomes, and the 80S ribosomes are divided into 40 and 60S subunits. and subunits are formed with the different proteins and RNAs. So already we discussed in the previously, and then these ribosomes are structurally participating in the transcription and translation, and especially translation. So translation is the place of the ribosomes. So in the production of the protein synthesis. Protein synthesis is done by the formation of the peptide bonds between the amino acids, which is formed the synthesis of polypeptide chain by the synthesis enzyme present in the ribosome. Then the major function of the ribosomes is the first function is the protein synthesis only, and then protection. So these two functions are done by the ribosome. And then finally we are discussing about the lysosomes. So lysosomes are the suicidal bags. These suicidal bags are morphologically four types are there. Primary lysosomes, digestive vacuoles, phagolysosomes, catalysosomes, and residual lysosomes. Then functionally, the lysosomes are taken about the four major functions are done by the lysosomes. That are histophagy, autophagy, and extracellular digestion and autolysis done by the lysosomes. And then centrosome. So centrosome is also another cell organelle. That cell organelle which is found out by Van Beneden, and the structurally these are the found in the center of the region of the cell and near to the nucleus. And these central body is made up with microtubules and lipids, tubulin, protein, and lipids, and enzymes contain these three important chemical substances. And functions of the centrosome is two functions are majorly done by these centrosomes. First one is the cell division, which is formation of the mitotic spindle, and which is participation in the cell division. And second one is the formation of the basal body, formation of the kinetosomes, or formation of the this basal plate of the cilia and flagella. These two functions are done by the centrosomes. So morphologically, showing the 9th pattern, and so this is the today class. We will see and we meet in next class and with a new topic. And until you have to go to the close of this session. Next we are going to the Zoom meeting. So shall we go to the discussion? I understood the class is useful but to you, and you may give the feedback of these 4 sessions. Please respond anyone before we are going to the close. So the time of the starting, 17 and 18 participants are there, but here now they are observing the only 4 more. 6, I think 4 students are there in the classroom. No problem at all. Thank you very much for your patience, and we will meet you next class.
Okay,
the
sir.
9th date of 9th. Then we will continue further syllabus. Okay, see you. Bye.